Guys, welcome to Talk with Nick. Today we have Professor Shelley, with whom we're going to talk a bit about plant biotechnology, cannabis, and also academic research and how we can include PhD or postdoc students in these kind of research work. So, Professor Shelley, thank you so much for giving me an opportunity to take your interview. Can you tell us a little bit about yourself and your work in the Carleton University?
Sure. I've been a professor at Carleton since 2005. I'm a plant biologist, and I study how plants grow and develop at the molecular level, so using techniques like genetics and molecular biology.
Because I read the introduction of your research lab. It says that you study the plant at the vertical aspect and the lateral aspect. So vertical aspect is the stem, and the lateral aspect we can talk about like the flowers, the leaves. So it's just like the human spine, right? Like the plant stem, we can call it as like the human spine, and the arms could be like the flowers and the leaves.
That's a great explanation. I love it.
Yeah. So now I was wondering how we can relate the genetics with the lateral aspect and the vertical aspect.
Oh, sure. Yeah, so all of the parts of the plant, it all comes from a collection of stem cells. And so what I actually study is how that stem cell population is controlled and how it builds those different parts, and that happens at the level of genes.
All right.
Yeah.
So, and my main reason to come and join you for the interview was because you were involved in the cannabis project, and you are using a strategy of polyploidization to increase the diversity in the speciation. So, what is polyploidization?
Okay. That's an easy word for, actually, it's a complicated technical word for chromosome doubling.
Okay.
Yeah, so the plant has one copy of chromosomes from its mom and one from its dad, and we just double it, so it's got two from its mom and two from its dad, and that just creates a lot of more genetic diversity, and the plants are usually larger, and there's various benefits that the industry is interested in.
Okay. And does this polyploidization happen naturally?
It can. Yeah,
Okay.
plants are great at this. Hybridization, yeah, it happens naturally, but we induced it chemically in the lab.
Okay.
Yeah.
Can you share some example where this polyploidization can happen naturally, like with disaster or with some fires, or if there is some floods, so the chromosome number can double in terms of stress?
Yeah, stress. That's
Okay.
exactly it. So sometimes stress will cause a defect when a cell divides, and the cells won't properly segregate the chromosomes, so one cell will get all of the chromosomes, the other cell gets none, and then that grows into a shoot, and the shoot produces seeds, and the seeds germinate, and then they might outcompete all the wild type neighbors and survive.
Okay. Right. And this now we have learned that during the natural conditions can also induce the polyploidization but in the lab we are doing it chemically, which is a little bit more effective and controlled. So what are the different kind of chemicals, or if there are any other lab-based studies to induce polyploidization?
Yeah, there's very traditional chemicals that people have used for years to do this, and they're basically chemicals that disrupt forming the mitotic spindle in a cell, and that's the thing that separates the chromosomes. So if you get rid of that structure, the chromosomes no longer divide properly. Yeah.
Okay, and if we have to explain this thing to an entrepreneur
Yeah.
working with the cannabis plant and wants to use this strategy to induce polyploidization in order to have, let's say, like a thick stem. If one have a thick stem, then we can extract the fiber for the cannabis plant. Or if we want to improve the lateral aspect of the cannabis plant, like having a big bud or having more sticky trichomes. So,
Yeah.
can we relate to those things like polyploidization and the characteristic of a cannabis plant?
Yeah, exactly. So when the plant has cells in it that have more chromosomes, the plant usually grows larger, and the gene pool is bigger, and that causes, say, an increase in the size of flowers that can lead to more trichomes and more medicinal oils, things like that.
Okay.
And also, the added benefit is that the plant, because it has a larger gene pool, might be better at resisting disease.
Okay.
So it's actually really beneficial.
Beneficial.
Yeah.
Cool. And when we're talking about polyploidization, chemical induced, and also we talked a little bit about the natural, the impact of the nature on the polyploidization. I was in back in September in last year. I did a expedition in the Himalayas for the cannabis plant. It was one of my friend's project. He's trying to study the diversity of cannabis in the Himalayas. So we found that as we were going from different region in the Himalayas, the plants were of different sizes. The soil texture of the mountain was quite different. The mountain slopes were different. So it was really interesting to see how the plants are different in terms of the smell, in terms of the leaf structure, the leaf color, the stem size. Really interesting. So is there any way that we could manipulate, or not manipulate, like copy the environment of the Himalayas in the lab and create different kind of plants? Because we know that the cannabis come from the Himalayas, right?
I'm just going to say that plants are incredibly adaptable. So when you see plants of different phenotypes, as they're known, varieties, growing on these different parts of the mountain, it's because they're adapted to that particular environment. So sometimes those changes are heritable, so they could be passed on to the next, daughter plants or whatever. And sometimes that's just the way of the plants adapting. So you can do that kind of thing in the lab.
Okay.
Yeah, it is possible. Yeah.
And how much money one will need to replicate that kind of environment in the lab?
I think it would take a lot of work.
Yeah. And it's only possible if the industrial and the academic collaborations happen, right?
Yeah, exactly. So the industry, they're interested in certain kinds of plants, and as an academic, I help industry reach their goal.
That's cool.
Whatever it might be.
That's one of the goal of me to create the content with academic researchers who are at the top level, also with the PhD scholars and the postdoc, because they are at the grassroots level and seeing different kind of problems with different set of eyes, and the academic researcher sees the problem with different set of eyes. So it's get interesting as I meet different kind of people. So now I was just wondering if you could give an example of polyploidization. How, if there is there are peer-reviewed studies if polyploidization has impacted the vertical aspect in terms of the stem meristem, or the lateral aspect in terms of improving the quality of flowers. If there is any studies published or some industrial product coming out of this thing.
Yeah, their industry is using this.
Or
Yes,
they're already using it?
They are using this. So in the plants that we created, there was changes, for example, in the terpene content. There were changes in the density of the trichomes on the leaves and things like that. And those particular strains, they might go to market. I'm not really sure. But one of the things the industry is really interested in is breeding, right?
Okay.
So if you take the polyploid plant and you breed it with the normal plant, you'll get a hybrid that's got an intermediate number of chromosomes.
Okay.
And it will be sterile, but this is very beneficial because the formation of seeds is something that reduces the quality of the product. So if you can make these sterile triploid plants, as they're known, that could generate a lot of money.
Okay.
Yeah, so that's one of the directions the research is headed.
And you talked about the plant breeding. How much time? Okay, let me rephrase this question. What the first step is the polyploidization. Then the second step is the plant breeding. How much time does it take for the polyploidization or the experiments related to polyploidization to be replicable and confirmed at the laboratory scale?
How much time?
Like if 3 PhD student is working on one goal, or even 1 PhD student is working, how much time a PhD student will take?
Well, it's actually not that time consuming, to be honest.
Okay.
I think what does take a lot of time is, I guess, creating the strains that you want to bring to market.
Okay.
Yeah. So creating the plants is not that hard. And in terms of human hours, one student could do this.
I mean, I'm now a lot of scientific questions are coming up, and I didn't want to go into that way. Just wanted to keep it general for young students, entrepreneurs,
Yeah.
and the cannabis enthusiasts.
Mm-hmm.
Okay, and what are the important agronomic trait with reference to the cannabis plant that one can solve using polyploidization?
Well, I think the most important thing is yield. So
Okay.
with the cannabis plant, of course, it's the medicinal oils that are desirable, and the polyploidation can increase the amount of those.
Of the total yield.
Yeah, that
Okay.
is the main thing.
And have we checked in the lab scale or in the industrial scale how much yield presently we can increase using polyploidization?
I don't know the answer to that.
Okay.
It might not be a doubling,
Okay.
but even say like a 10% increase would be great.
Oh yeah.
Yeah.
True. And how it will affect the cannabinoid profile, considering that we know that we are assuming that there are more than 120 cannabinoids, or it could be more. We don't know the exact number,
Mm-hmm.
but can we use polyploidization to manipulate the overall composition of the cannabinoid profile?
Absolutely.
Okay.
Yeah.
And another question that I have on this is that considering that the cannabis is not the, I don't know if it's correct, but if cannabis is not the native to the North American land,
Mm-hmm.
it's native to the Asian land,
Mm-hmm.
and it grows in a large diversity over there, so the cannabinoid profile will be more complex, or most of the plants over here are hybrid, if I'm not wrong.
Yeah.
So how polyploidization will be able to change the cannabinoid profile in the hybrid plants?
You can't predict these things.
We cannot put, okay.
No, you can't really predict, because when you increase the gene pool in a plant, there's a lot of interactions that take place, and it's very difficult to predict the outcome. Luckily, it's pretty easy to make these plants, and then you can just check. Yeah. So it's all about, breeding, what plant you start with, making a few varieties, and then checking.
Sure.
Yeah.
Okay, and another question is, now the questions are related to, like, there are a lot of cannabis entrepreneurs who are popping up in the market.
Yes.
And it's just like when they work with the cannabis plant, mostly they think about the large yield.
Mhm.
And as they will go more into detail, it will be the cannabinoid profile, and when there will be more research about cannabinoid profile impact on this disease, this health, or this aspect, it will change. So is there any advice or the suggestion that you can give to cannabis entrepreneurs on how to investigate into this area?
I think it's important to know the market and to have a goal.
Okay.
Yeah, this is what I think is the vision.
The vision.
Yeah.
Thank you very much. It was really interesting to talk about the cannabis and the polyploidization. I was just checking on internet what is happening in the cannabis industry, and your name came up, and I thought of contacting you. So it was really pleasurable to talk with you. I got to learn many things about this subject. Then on another aspect, I wanted to bring the attention, because a lot of my viewers are students or enthusiastic students or academic who wants to enter into the PhD or master's based research. So if you have any suggestion, what do you look into a student profile when you are selecting him or her for a PhD project or a master's project?
That's an interesting question. I look for a student that I think would be a good fit to the project. Are they genuine? Do they have a good track record? And do their references have good things to say about them? So when you put all that package together and I have a little chat with them,
Okay.
that's
That's all.
That's what you want to
So,
do.
is there any would you see that they have worked on exactly the same kind of project, or it's just they are curious and
There
open-minded?
you go. Curious and
Okay.
open-minded. Have good training, good work skills, good communication skills.
Thank you very much, Prof Shelley. It was a pleasure to talk with you and giving me an opportunity to discuss on all these subjects. Thank you very much.
Thank you for having me on your podcast.
Thank you.